Imagine there was this book in the library that was super rare and you wanted to use it for a research project, but they don't print this book anymore. If you go to the library, they're not going to let you take the book. Therefore, if you need something from that book, you're going to have to read it in the library or you're going to have to make copies of it. And all you can take out of the library is the copies because they need to protect that book. Well, the same thing happens with DNA. Since there is only one copy of DNA for the entire cell, that DNA needs to stay protected inside of the nucleus. So if a cell needs to know how to do a specific job, it's going to need a copy of a specific gene from that DNA. That is where protein synthesis comes in. So it starts off with step 1, messenger RNA. They come, and this is a single strand of genetic material. So this is going to come and get a complementary copy of a gene, and it is going to take that message out of the nucleus over to a ribosome, and on that ribosome you have transfer RNAs that are going to bring amino acids to that messenger RNA and build a polypeptide chain, which will later get folded into a protein. Well, how do we know which of these codons, a group of 3 of them, how do we know which amino acid that stands for? Well, the transfer RNA is going to actually have a complementary code called an anticodon that goes with the codon. So if those two match up, then it knows, all right, I have the correct amino acid. This is what I'm going to leave behind. So if you look at an amino acid wheel, it's going to show you which codon represents which amino acid. And there's about 20 different amino acids. So on this messenger RNA strand, there's usually always a start codon, and it's AUG. And I remember that because school starts in August, and the reading of the code starts with AUG. And then there's a stop codon at the end of every messenger RNA strand, which is one of these 3 right here. So we're going to play a little protein synthesis bingo to further understand how this process works. I'm going to come on over here, and I am going to turn the nucleus over here, which is the bingo wheel, and see what our first sequence is going to be. G, T, T. So according to our matching rule right here, A goes with T and C goes with G. The only difference is when you're talking about RNA, you're actually going to have a U instead of T. So keep that in mind. So if our DNA sequence is GTT, that means the mRNA sequence is going to be CAA. Now, what amino acid would that be? You always start in the middle here. So start with C, and then go out to A, and then go out to A, and it is glutamine. So we have to find glutamine in our pool of amino acids over here, and I have found it, glutamine. So the tRNA is going to bring glutamine over here and leave the amino acid behind. So let's get a couple more of these just so you can get the idea here. So the next codon in the DNA sequence is CCA. So let's find out what the mRNA sequence would be. GGU. So let's look on our wheel and see what that would be. GGU is glycine. So let's look over here, and I see a glycine floating around in the cytoplasm. So there's our next one. And now we have 2 amino acids. So those are going to link together with a peptide bond and form a polypeptide chain, which, by the way, would be a really cute name for a dog, is polypeptide. Anyways, you get the idea. So they're going to link together there. And let's just do one more here. C, G, C. Okay, so that would be G, C, G, which is G is alanine. So we would come over here and look for an alanine. I see one. And we would be working as a class to do this. It's more fun in teams. So now we have our amino acids, and they all have to be linked together in this peptide chain to make the protein. So the last step, why are these not going together? There we go. So the last step will be to take that polypeptide chain that was created and fold it up into the protein that's needed for whatever its specific job is. So that is how protein synthesis works. I hope that's helpful. And then at the very end, we would have a stop codon. One of the stop codons is UGA. So I like to say, don't UGA believin'. Don't stop believin'. Have fun with your protein synthesis lab.